Good morning, everybody. Let's wait a couple of minutes till people roll in. In the meantime, if anyone has any questions about anything that we've been covering, feel free to shout out. Plenty of time. All right, let's get going. Okay, so we have not that. Not yet anyway. Um, we need to finish up this, so get the chat up. All right, so we finished up with variable expressivity and penetrance, I believe. Is that right? We're talking about mitochondrial disease, or was that a different class? Remind me where we ended up on Wednesday. Yeah? Okay, good. Oh, that's right. Yeah, because I didn't teach on Thursday. That was an invited lecture. It's hard remembering what's going on, to be honest, sometimes. Okay, so let's get rid of this. Dismiss, because I'm in it. Right, so essentially, variable expressivity and penetrance is the idea that for the same genotype, you can express a different phenotype. Right, so even if you have the same mutation, depending on other factors, in the case of mitochondrial disease, like number of diseased mitochondria, what tissue they're in, stuff like that, you can have a different phenotype, a different expression of that genotype to someone else with the same genotype. Okay, and so that leads us quite nicely onto this idea of complex inheritance, quantitative inheritance. And so really when we're talking about complex inheritance, talk about two different things. One, polygenic traits. All right, I'm going to write that up. I lost my chat box again. And that's basically more than one gene controlling the trait. Pardon me. Or trail. Yeah. And the second one is gene by environment interactions. Right, essentially that's the whole nature versus nurture debate, and really it's kind of not a debate because it is both, right, for almost all of the things that we can see. So the kind of the revelation for a lot of students is really that almost all human traits are polygenic. Eye color, polygenic. Widow's peak, not even really a trait. Tongue rolling, which I can do, that's almost certainly polygenic, right? And so one way you can do that, figure that out, is by looking at family histories, right? We're talking about before inheritance, and it's quite easy to figure out which traits are single gene recessive and which ones aren't. And also just it kind of makes sense from our biology, right? So we're really complex organisms. We have an awful lot of different systems controlling different aspects of our biology and behavior. Oh, sorry. And also, it takes an awful lot of time even to become a functional organism. It was about 7.5 months, give or take, of development in utero. And so it's kind of makes sense, really, that we're going to have multiple genes controlling any given thing, right? And so a lot of the stuff you're taught maybe at high school, maybe even Gen Bio 1, if they still do that stuff, is wrong, right? So all kinds of different examples. I can't even remember all of them. That attached earlobes. Eye color is the classic one. Eye color is controlled by at least 3, I think maybe 4 different genes, right? Which again makes sense because we've got pigment production, we've got pigment getting put into different cells, we've got different colors of pigments, all kinds of stuff. So however you think about it, it kind of makes sense. And also, I'll get to gene bioenvironment interactions in a little bit. Come on. One of the classic signs of a complex trait, right, is a normal distribution. And so if you say, for example, measure height, remember that experiment we did at the beginning of the semester in lab? If we did that with an actual real sample and a big enough one as well, and plotted out how many people were each height, you'd get a normal distribution. You'd get that kind of classic bell curve, right, where most people are in the middle, essentially, right? So that's a classic telltale of a polygenic trait. Also, we've got gene bioenvironment interaction, right? And so this doesn't have to be for single gene traits. It can be for polygenic traits. Classic example is height, right? So height is a polygenic trait because, let me just get my chat box up in case people ask anything. Yeah, height is a classic polygenic trait because during development, you've got genes which control your body plan, right? And those vary slightly, not a lot because we all have the same body plan, but certainly how long your body is varies. We've got genes which control bone growth. We've got genes that control muscle growth. Pardon me. We've got varying levels of growth hormone, which control overall growth of organism. We've got genes involved in metabolism, right, which provide the energy for that growth, for those rounds of cell division, which generate those structures, right? So we've got multiple different genes involved in just one single trait. Now, if you think about height of, I don't know, individuals from Bangladesh, maybe, right, which is next to India. Typically, it's a very poor country, malnourished. We could think about Syria, right, which is in the midst of a civil war. More the classic sub-Saharan Africa, which undergo periodic famines, right? And then compare those to, plump and well-fed Americans or Northern Europeans, British people, right? You're going to see that there's going to be a difference in height between those two populations, right? Now, if you take, so one explanation would be, well, I mean, their genetic background is different, right? So it's down to their genes. But if you take individuals from that malnourished environment and put them in a well-fed environment, they will attain similar heights to the people that are in that well-fed environment, right? So it's not just genetic, it's also based on the environment too. And you've also got things like this, where you have basically different responses to their environment, right? So you can have, particularly in crops, right? You can have crops which do pretty well in all kinds of environments. They're very robust, right? So You know, things like kind of different types of potatoes or wheat from more subsistence kind of agricultural environments. And then you also have strains which are like super highly tuned and bred to be really high yielding in very good environments, right? That would be things that you'd find in the monoculture out in the Midwest. Now, if you put one of those highly tuned plants in a flood situation or a drought or high temperatures much higher than it's used to, it tends to perform very poorly, right? Because it's much less robust. Essentially, it's a trade-off between in plants, typically crops, yield and robustness, right? So the higher the yield, the less likely that plant is able to survive adverse conditions, right? And that's actually a really interesting thing from our perspective in climate change, right? So the climate is changing in all kinds of different ways. And one of the ways will be changing adverse weather events, right? So there's an awful lot of work being put into figuring out ways of adapting plants to different conditions, more saline conditions, you know, because sea level is going to rise and encroach further inland. You know, drier conditions in some parts, wetter conditions and other stuff like that. Okay, so that's pretty much that from like the classic non-Mendelian stuff. Does anybody have any questions before we move on? Don't be shy. You can ask me anything. Honest. Had someone in lab the other day said, I know I want to ask you a question, but I'm not sure what it is. And I was like, well, you know, ask me what my favorite color is. What's my favorite food? So that got things moving. Main points that'll be on the exam. Well, all of it. It's hard to say specifically because, to be honest, I can't remember. But you'll need to understand essentially what are the key markers for all of these different things, right? So incomplete dominance, eek, come on then. Classic sign of incomplete dominance is both an intermediate phenotype and a 1:2:1 ratio in the F2 generation. So that's the key thing. Codominance is expression of both phenotypes, both the dominant phenotypes. For example, complementation for just from this. What I mean by variable expressivity, right, which is the variation in severity, versus variable penetrance, which is variation in who shows the trait. And also the idea of Benji's outside, love. The idea of this kind of normal distribution for complex traits, and the fact that most traits are complex. So that's kind of really the main points from this particular PowerPoint. Obviously, the exam will be on multiple PowerPoints, right? So you need to do that for all of them. But yeah, good question to ask. And again, from I haven't actually looked through the grades for both sections yet. I think I did it for the other one, which actually was all right, but I haven't done it for this one yet. So you now know, you have a pretty good sense of like my exam style, right? So just as with the first exam, there will be a mix of basic recall stuff. There'll be a mix of working things out. There'll be a mix of kind of going from one thing to another, right? And so, for example, one thing that you'll have to work out on the exam will be linkage, right? So just as with the other exam, it was chi-squared. right? So that was the chi-squared and probabilities and stuff like that. This one of the main things you're going to need to work out will be map distance, right? So this is something that you want to practice. And we've got this and next 2 lectures to practice this stuff, right? And so just in general about success in this class, obviously you have to do the homeworks and the do well in the lab, right? Those things you can't afford to pass those up, because if you do, then you'll probably not be successful, right? But you also really do need to maintain as best as you can, a good level in your exams. And the only way to do that is to study, and to study constantly. I'm not talking about studying hard for a couple of days before the exam. Pardon me. That's simply not enough. Unless you have a particularly good memory and you're naturally very good at this and understand it anyway, which some of you are, but a good chunk of you won't be, right? I'm not that kind of person either, to be honest. You need to work on this constantly, right? So after today's class, right, you need a good half hour to an hour to review what did we cover, right? Let's write down the different things that I need to know, and I'll see how well I know them, right? Every single class you need to do that, and then just study on top of that, right? We're talking about reading through the PowerPoint before coming to class, right? So you have an idea of what you're going to be looking at, and it's not all going to be fresh first time. How many of you have already looked through the linkage PowerPoint? Is it anybody? Don't worry about saying yes or no. I'm not going to judge you. Okay, so this is a really complex topic, right? And it's a topic that students really struggle with, right? And that's why I've assigned so much time to working on this. But there's a limit to how much time I can spend on it, because we've got to cover other topics too. And that's only going to be what? Oh, would that be like a couple of hours, probably, give or take, going by what we've got left in this class. And that's not a lot of time to learn something from scratch and be very good at it, right? So this bit with me, I'm kind of giving you pointers on explaining tricky bits and kind of helping you through stuff, but I can't do the bulk of the learning. That really is on your shoulders. And that's the same for any class, right? Not just this one, but any, particularly as you kind of go forward in your degree and start taking more challenging classes, upper division classes, right? So this is just a habit you need to get into. You need to do this constantly. Read through the PowerPoint before class, right? Circle things that don't make any sense whatsoever. Those are things you're going to pay special attention to in class, right? Maybe you do understand something but don't quite get like the relevance. Write that down as a question, right? Asking questions is how you get better, right? It's how you clarify points, right? Don't expect to be able to understand all of this yourself. That's what I'm here for, right? That's my job, is to help you, right? It's not, in many ways, it's not actually to teach you. In many ways, it's to help you understand the points which you can't learn on your own. Otherwise, you just go to Wikipedia and you can find some professor's genetics schedule online, and then you just teach yourself, right? I mean, you wouldn't need me. I would be superfluous, right? So really, that's kind of what I bring to the table. I know the stuff that you're going to struggle with because I've been doing this for a while, right? Some of the things I've struggled with too, right? It's not that just you alone. And so those are things I can help you with, but you've got to pull an awful lot of weight yourself, right? And so typically the students that do well in this class aren't Sometimes they're the ones that are smart. I mean, that's just kind of the way it goes. But almost always it's the students that are well organized and work really hard over a long period of time on this course, right? And those are the students that are always successful, right? Those are the students that get some of them get A's, not always, right? But they'll get B's and they'll definitely get C's, right? This is something that you have to make almost your full-time job, right? And that's just every every course that you do is essentially its own full-time job. And so that's really my best advice in terms of succeeding in this course, right? Make this your full-time job. If you can't because you have other stuff going on, hopefully none of you have overloaded on courses, because this is one of the worst courses to overload with, right? But I know that you will have other kind of drags on your time. One thing is to try and figure out how those can be reduced, right? And this is a really difficult kind of conversation to have, right? Because I certainly wouldn't want to tell you how to lead your lives, because that's not my place. And I don't know how you should lead your lives, because I don't know you well enough anyway. Even if you were my kids, I probably wouldn't be telling you, because they would just piss you off, right? So I'm not going to tell you what you should do, but it may be worth, if say one of the drags on your time is having to look after children, right? Is there a way that you can enlist the help of friends or family to take a little bit of load off you, right? Kind of, I don't know, kiddie pool in a sense. I'll take your kids for a couple of hours so you can study if you'll take mine for a couple of hours, later on in the week, that kind of thing. If you have work, right? Figure out if you can afford to work one shift less, right? Or work in a different time or something, right? Just so you can put aside a time, right? It may be that you just can't watch. I don't know. My wife's an American Horror Story. It's a horror story, yeah, something like that. Maybe you won't be able to watch as much of that as you want, right? But it doesn't last forever. It's just a semester, right? And once you get through it and you get your C or better, then you're good. You don't have to take this again. You can move on to something else, right? But this is something you just have to spend a lot of time on, right? And there's no real kind of way around that. I remember many, many years ago doing my degree. I pretty much worked 14 hours a day just studying. Sometimes went out, had a few beers, and did other stuff, but most of the time I was studying, right? And it worked. I got the degree that I wanted, and I got on to the next stage of my life that eventually led here, right? And so there is some degree of sacrifice involved. It's very difficult to have everything you want and do well at everything that you're trying to do, right? So I'm not going to tell you how to do it. If you want to ask, and we can do this, privately, obviously, either by email or by Zoom. And I'm more than happy to arrange meetings with any of you to talk about stuff like this. I can give you some advice. I certainly, again, wouldn't imagine telling you what to do, but I can, give you some tips on how to manage the load, maybe, right? But this is something that you have to really commit to. And if you've done poorly in the first exam, that happens. That's fine. You can afford that. One of them. You can't afford to do poorly in your second exam, because then you start just fighting against simple arithmetic, like trying to catch up back to that level that you need to be able to get a C. Right? It gets ever harder the further, down the road you get towards the final exam. All right. So yeah, that was a somewhat unintended pep talk. Well, not pep talk. I don't know what kind of talk you would call it. Talk. But yeah, and it's difficult, right? I mean, sorry, shouldn't have muted myself there. I mean, one way of thinking about this is that, a standard degree is 4 years, right? May take you a little bit longer than that, but typically 4 years. And for 4 years, you might have to just suck it up and do things that you don't want to do, right? Hang out less with your friends or family, you know, uh, go on fewer holidays, watch less TV, do less of your hobbies, whatever they might be, right? But it's only for 4 years. And then you get a piece of paper, which, you can take to an employer or to a graduate school or wherever and say, look, I can suck it up and I can do my job. Give me a job. And that might help you get where you need to go next, right? And it's just 4 years. I mean, I'm over 40, so that's 1/10 of my life. My degree is actually 3 years long, because I didn't have to do all the kind of non-science stuff that you lot do. Uh, but again, it passed. I still had fun, enjoyed it, but I got what I wanted out of it, and I got to the next stage of my life to do what I really wanted to do, which is doing science. So there, there is a cost, right, in being successful in this stuff, because otherwise everybody would be doing it, and it wouldn't be worth doing. So think about that in your own individual context. Think about all the things which are uh, impeding your ability to be successful in this class and others, right? Because almost certainly you're taking probably a chemistry class at the same time as this, which is also another, I don't know where you'd be, Gen Chem 2 maybe, or Go 1 perhaps, depending on where you're at. OChem 2, holy shit, I would not want to do genetics and OChem 2. Well, good on you anyway. But it's really, really hard, right? And it stays hard. You're not going to have these core courses, perhaps, but you'll have other upper level courses like molecular biology or microbiology or maybe even developmental biology if I ever get to teach it again, you know, that are difficult. Yeah, I don't think, and there aren't very many people that like OChem, to be honest, or chemistry in general, which is unfortunate for the people that have to teach it because they're really good at teaching it. But they never get any chemistry majors who really are interested in it. But anyway, those are just, again, great examples of things that you just have to do well at, and you just suck it up and grind your teeth and you get on with it and do it, you know. But also another general piece of advice before we get onto this, ask me questions, right? Do not try and learn all of this stuff yourself. That's what I am here, is to help you. And there will be things which just kind of don't make sense. And it's only through you asking me questions that I know what those things are, and then I can help you with them. Had a great one in lab, trying to write out genotype of stuff and recombination, and someone put their hand up and said, I just don't understand that. So I drew it vertically, made perfect sense, right? So you don't have to figure all this out yourself. Okay, so what I want to do over the last 20 minutes or so, other than listen to my puppy trying to chew his way through the back door, is I want to talk about linkage. So the idea of linkage is something that breaks Mendel's third law. So the law of independent assortment. And so Mendel, through either luck or guile, I'm not sure which, chose all of his traits, his P traits, that were on separate chromosomes. And he chose 7, or he's reported 7. And funnily enough, anybody guess how many chromosomes P's have? They got 7 chromosomes, right? So when every trait is on a different chromosome, doesn't matter what trait you look at, you'll see that the random orientation of chromosomes on the metaphase 1 plate and also the segregation of alleles generates all possible gametes in equal frequencies, right? So essentially, if you have two genes and the two individuals are heterozygous, you'll get a 1-to-1-to-1 ratio of dominant, 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 recessive, recessive, dominant, recessive, recessive, like what you see down here, right? However, in real life, IRL, sorry, my kids always tell me that, and I'm trying to sound, hip, I guess, or even just saying that. No one says that anymore. Oh well, never mind. I'm doomed. So in real life, you have chromosomes that look like this, right? This is salamander for some reason. This is human chromosome, right? These aren't even genes, right? These are linkage groups. Oh, I'm sorry. Haven't had my coffee yet. Right. I can't remember actually on chromosome 19 how many genes that chromosome has. I don't know. I'd say at least a few hundred, 200 or 300 at least. I can't. Chromosome 19 will be one of the small ones because they kind of rank them in size. But there'll be several hundred genes on here, right? And so if you're looking at traits that are controlled by genes that are on this chromosome, then you'll not have independent assortment of all possible gametes. And the reason is that you have to rely on recombination to separate those different alleles in meiosis, right? To get that jumbling of alleles. If they're very close together, that might not happen and they'll travel together, right? So essentially, linkage is all about when genes are on the same chromosome, they're physically close to each other, and recombination does not always happen between them, right? And so basically, this breaks a second law? Oh, I thought it was his third law. Anyway, this breaks Mendel's law of independent assortment, right? Because what allele you get for one gene will affect what allele you get for another gene, right? Stop me at any point if any of this doesn't make any sense. All right. Sorry, every time I click on the PowerPoint, my chat box goes away. Zoom is a pain in the backside sometimes. Better than WebEx, though. That is a complete horror show. All right, so essentially what we mean by recombination, right, is this crossover, chiasma, between non-sister chromatids of the same homologous chromosomes, right? So if you remember, actually, let's go be down here somewhere. All right, so basically going back to this, actually, is this a better one? No, that one sucks. Here we go. This one's better. Yeah, so going back to meiosis, right? This is interphase before any splitting up of DNA happens. And we go from individual chromosomes, right? So this is a homologous pair. Each one of those gets copied, and then they stick next to each other during prophase 1, right? So during the condensation phase of meiosis 1. And then in the process, they swap over bits, right? And remember, that's what actually holds those homologous chromosomes together during the beginning of meiosis 1, right? Up until and including metaphase 1. And if you remember, it's the glue at the ends of the chromosome that gets destroyed, which allows those chromosomes to separate during meiosis 1 and reduce the chromosome count, right? So the ploidy, going from diploid to haploid. right? So that's kind of what's going on. That's what we're talking about, this recombination, which before we've really kind of concerned ourselves with how that, holds those chromosomes together, the actual mechanics of it, right? What we're going to do now is actually look at what effect that has on what alleles you get. So in a sense, it's kind of Mendelian inheritance in a way, but also how you can use that as a tool to figure out how far apart those genes are. And that is exactly what you're going to be doing with the fly linkage project, right? So that's, and if any, it's kind of hard to broach the subject, but if you have any of you that are not in my lab section have questions about that fly linkage project that Mr. Penny is not able to answer, please do let me know and I can help you with that, right? So that you get the full benefit from that experiment. And that's not because Mr. Penny doesn't know what he's doing, it's just because he hasn't necessarily taught this a bunch of times before or done that experiment. Right, so essentially what we mean by recombination and crossing over, right, is that these non-sister chromatids, right, so they're actually on one of each different homologous chromosome, not different homologous chromosome, like one of the pair, right? They're very close to each other. During prophase 1, they're lined up perfectly. And we'll actually get into the mechanics later on, but they swap bits of chromosome. So if you look at the original parental chromosome up here, it has 2 recessive alleles, right? We use letters for recessive alleles. And the other parent has 2 wild type or normal alleles, right? These are used, represented with pluses, right? This is the kind of language that we'll be using, we have used in lab, or at least in my lab, and you will be using throughout this, right? So it'll look something like this, right? If you're writing what we just looked at above, you would write it like so, right? Plus plus being the wild type chromosome, WM being the chromosome with the 2 alleles, mutant alleles on them, right? So if we're using like the old style Mendelian way of doing things, these would be capital letters, these will be lowercase letters, essentially. Here we go. So when you have that crossing over, right, and you go through the whole of meiosis to the end of meiosis 2, you'll end up with 4 different chromosomes. You'll have a parental chromosome here, right, which looks just like the one up there. You'll have another parental chromosome here, which looks like this one just here. You'll also have 2 non-parental chromosomes, or another way of putting it is 2 recombinant chromosomes, right? And these are chromosomes which have a different combination of alleles to their parents, right? So I'm just going to write that down because that's important. So the parent, dang it if I can spell right. I mean, parental chromosomes is easy, right? It's same combination of alleles as what you see in the original pre-recombination chromosomes. And they're called parental chromosomes because those are the chromosomes you got from your parents, right? And so in each of your cells, you will have essentially a maternal and a paternal chromosome, right? And so when you go through meiosis and generate gametes, those are then what are called the parental chromosomes, right? Obviously, in the next generation, what they call parental chromosomes will depend on which ones they got from which parent, right? When the non-parental, and these are also called recombinant, These have a different, oops, if I can spell that right. These have a different combination. So this is actually good pointer here. You have to be very, very careful not to make assumptions about what you are seeing in terms of allele combinations. You cannot always assume that the parents will have both mutant alleles and both wild type alleles, normal alleles. You cannot assume that. They could be one of each, right? We'll talk about that in a little bit, what that's called, right? Could be one recessive and one wild type, or one dominant, depending on, which way around you are, right? And so the recombinant chromosomes will then look like these ones. They'll have either both dominant or both recessive. They're not parental because they don't look like the parental chromosomes, right? So you can't make any assumptions, really, about what chromosome has what combination of alleles. The only way you can look at this is by looking at the numbers, right? And this also goes for gene order in 3-point crosses. And I would strongly recommend you have a look through this PowerPoint after we talk stuff that we haven't covered yet, right? You cannot make any assumptions about it. You have to kind of go to like, and just look at the data and work it out from scratch, right? If you make any assumptions about what has what, then you may end up being wrong, right? And that would suck, obviously, if it was an exam. However, right, and this is, sorry, something that I talk about in the lab. This assumes that you're going to have a crossover event between these two genes. Now, these two are way far apart. These are like, I call fish this big, kind of far apart, right? And as you can see from here, come on, if I can get to it, you can have genes, or these are linkage groups, but imagine these are genes, right? You can have genes which are both down on one end of the chromosome. And so the likelihood of a crossover event always occurring down there is just the same as a likelihood event of that crossover occurring up here, or here, or here, or here, because crossing over is random, right? So where this thing occurs is random. Every time you go through meiosis, a full round of meiosis, oops, put that back there. It could be down here. It could even just be out on the tip down here, presuming that this isn't right at the very end, but up against the telomere. Could be over here. Maybe it doesn't even occur on this arm of the chromosome, but that's pretty rare, right? So you can actually have a situation where you have all parental chromosomes from one round of meiosis. Now that, and again, this is in reference only to these two genes. Obviously, these ones in the middle will be non-parental because they will be, you will have recombination occurring. You'll have recombination occurring here on this arm. You might have recombination event occurring here on this arm, but you will not see those. You won't see those because you're only looking at these two genes and their phenotypes. Does that make sense? So recombination always happens. It just doesn't always happen between the two things that you're interested in. And essentially, if it doesn't happen between the two things that you're interested in, you won't see it, right? Unless you're, I don't know, sequencing the chromosomes every time, which just you wouldn't do, right? Now, other times, again, just by random chance, right? So the probability of recombination occurring is largely the same, give or take, all the way along the chromosome arm. Doesn't happen at the centromere, doesn't happen at the telomeres, right? The very middle and the very end of the chromosome. But it will happen in between randomly. And so other rounds of meiosis, you will have a crossover event, right? And so you will have recombinant chromosomes or non-parental chromosomes appearing in the next generation. Now you can see, okay, so assuming that this is the case randomly for every random meiosis, if you were to get a collection of gametes and use those to fertilize the next generation, would you see equal numbers of individuals with parental and non-parental or non-recombinant and recombinant chromosomes? What would be the relative frequency of these ones down here? Would it be the same as the others? Question to the class. The answer would be no, right? So you're essentially what you're looking for, if linkage is occurring, is you'll be looking for two combinations of alleles, two genotypes, or even two phenotype combinations, which are present at a much lower rate than you would predict by independent assortment, right? And so that is the sign of linkage, right? Is that you'll get this these other combinations at a lower frequency than one would expect if those genes were not linked, right? And so if this were if these genes were unlinked, right, they're so far apart, that recombination always occurs, or they're so far apart they're on different chromosomes, right? One's over there and one's over there, right? Then you would expect a 1-to-1-to-1 ratio of these four different combinations, right? However, if they're close enough that meiosis does not always happen, or not meiosis, recombination does not always happen between them, you'll get more of the non-recombinants, the parental chromosomes. You won't see these ones in the middle, right? And so overall, you'll get an excess of the parentals, the non-recombinants, or a less than expected amount of the recombinants. Anybody got any questions? Okay. And so this is, come on, here we go. You'll see here. So these are two traits which are, well, we don't actually know, right? So if you kind of cover up the bottom part, the F2 generation, right? The P0 and the F1, seem to be behaving according to Mendelian principles, right? So we have homozygous, well, presuming purple flowers and long pollen are dominant, because that's what we see in the F1 generation, right? So we've got two different traits, flower color and pollen shape. And we're seeing them separate here, and we see the heterozygotes here. However, you would presume if these were assorting independently, right? If there's no effect of one gene on another, then you would expect a 9-to-3 to 3-to-1 ratio. Hello, love. Hey, sweetheart. Right? So you'd expect out of that, if there was, I don't know, 284, you'd expect a third of that. Yeah, that's right, which would be, I don't know, what, 90 maybe, something like that, right? Of the intermediate, the mixed phenotypes, right? So the purple and round, or the red and the long. However, what you see is far, far fewer than that, considerably fewer than that, right? And so this is showing you that the parental combinations of alleles are present at a much higher rate than the kind of the mixed or the recombinant, the mixed combinations, right? The ones that you don't see in the parents, the non-parentals. And so this is a classic sign of linkage. This isn't epistasis, right, which we talked about in non-Mendelian inheritance, because we're not masking one or other of these phenotypes. You still get the 4 phenotype combinations as you would in a normal Mendelian dihybrid cross, but you're not getting the numbers that you should be. This is the classic sign, essentially, of linkage. Okay, so that's enough for today. Woo-hoo. And we'll continue this on Wednesday, right? So we've got all of this stuff to go through. And what we're so there's a lot left. My aim is to basically get through most, if not all of this on Wednesday, and then spend Friday doing problems, right? We'll do some problems during this to illustrate the point, but I'll make up my usual random ass bunch of phenotypes, which don't make any sense. I don't know, like glittering unicorns or something like that. And we'll actually work through a bunch of linkage problems to get you practice that. The same as we did with the chi-square stuff, we'll do it with this so you get competent and proficient, okay? And that will be on Friday, right? So please do look through this. Don't expect yourselves to understand all of it, and that's fine, but it will kind of give you a heads up of what's coming. All right. Thank you very much, everybody. I mean, repetition is the key here, JM. You know, and don't expect for everything to lodge properly in your brain after just a lecture. It's a lot of stuff to cover, and not all of it will go straight in, right? So going back over this is the way to pick up on that stuff and make sure it sticks. Or if it doesn't stick, that's a perfect question to bring to me and say, hey, why does this work like that? Right? And I'll be able to look at it in a different way and help you understand. All right. See you all on Wednesday.